MSPC Keynote. So Dr Pitcher is a Senior Research Fellow in the Department of Medicine, University of Otago, Christchurch, and holds a PhD in Neuroscience from the University of Otago in Dunedin. For the past 16 years, Toni has been involved in a number of projects relating to Parkinson's and dementia, including the New Zealand Brain Research Institute's longitudinal Parkinson's cohort study, where she coordinates the collection, storage and use of biological samples. These samples are used in conjunction with research collaborators, and they can be local, national and, interestingly, international collaborators, to help further our understanding of biological pathways involved in the disease processes through the investigation of genetic and epigenetic factors and the investigation of blood protein levels as potential markers of the disease. Recently, Toni received funding from the Neurological Foundation to look at trends in treatment, mortality and incidence of Parkinson's. In this project, Toni will use health administration databases such as medication prescriptions to provide up-to-date numbers of people with Parkinson's and also identify trends in treatment. She will also use information from death certificates to identify the main contributors to death in this group. And this information is really useful for healthcare, for support services, and for researchers to help improve the care of people with Parkinson's. And actually for MSPC, it's really helpful for us to know how the numbers are looking in Canterbury as well. Today's Toni's talk will cover insights from NZBRI to Parkinson's cohorts, NZP3 and NZPEGS? NZPEGS, from the beginning of the disorder, environmental and genetic risks, to the main causes of death and her plans for future research. Join me in welcoming Dr Toni Pitcher.
Hey, yeah, here we go. Well, thank you very much for that welcome, Amelia. It was very thorough. I had a moment where I was like, she's talking about everything that I'm not going to talk about today, but we did get there in the end with a little bit of overlap. So yeah, I'm going to just use our two Parkinson's cohorts that we have at NZBRI as a little bit of a platform to talk about some of the things that we've done over time and focus in on epidemiology and mortality associated with Parkinson's. And I can tell you that you guys are the first to see the mortality data. So it's a little bit of a work in progress, but we've made some really good progress just recently, and hence why I've included it today. So I'm going to start off with this, which is sort of the stereotypical timeline of Parkinson's disease. And what it shows is on the top part of the figure, we have our motor symptoms. So things like tremor, rigidity, and bradykinesia, which is just that fancy term for slowed movement or reduced amplitude in movement. And down the bottom of this timeline, we have what we call the non-motor symptoms of the disorder. So these are things that over time have become more and more obvious that these are just a core part of the disorder. And so for those of you who are sitting on the timeline, I do acknowledge that many of you are on this timeline somewhere, or if you're supporting someone on this timeline, you'll see some things that are familiar to you. You might also see a few things that you've not come across yet. So I think one of the key things is there's a lot up there, and everyone gets their own combination of those symptoms, okay? So just because it's up there doesn't mean you're going to get it. And also, I think too, the place on the timeline might be different for some people as well. So you might get it something a little bit earlier on in the timeline compared to someone else. There's a couple of key things that I like to point out on this slide, and that is this here, although I did delete my big red arrow, is the point of diagnosis. So Parkinson's is still a clinical diagnosis. It requires a medical professional to observe the patient in front of them and identify some key factors. And that is these three motor symptoms which I mentioned. So everyone with Parkinson's disease must have bradykinesia. So if you get your index finger and your thumb, and those of you with Parkinson's will be familiar with this, if you can tap them together and maintain a nice rhythm and a good amplitude of movement, you're probably doing okay. But with Parkinson's disease, we know that people slow down the movement, the amplitude of the movement decreases, and sometimes it's very difficult to actually get a gap between the index finger and the thumb. So that's the easiest way that we can test for bradykinesia. It's obviously not the only form of it. So everyone must have bradykinesia, and then you must have one of these other two. So either rigidity or tremor, or you could have both. So the diagnosis requires bradykinesia plus rigidity or tremor. Obviously, it's not that simple, because we know that even at the time of diagnosis, there's probably going to be a number of other things present as well, and that's where some of these non-motor symptoms come in. And you might remember the doctor asking you lots and lots of questions at the time of diagnosis about lots of different things, and you're like, why are they asking me that? That's got nothing to do with this. Well, it probably did. And they may have also asked you about things that may have happened a long time in the past as well. And you'll note that at the time of diagnosis, these motor symptoms have also probably been there for a wee bit, maybe 5 to 7 years max. They would have been very slight. They were probably at the point where you could ignore them, so you didn't feel the need to act on them immediately. But obviously over time they've increased, because this is a progressive disorder, and you've got to a point where, okay, I need to get this checked out. If you look at the bottom part of this, you'll note that this non-motor timeline extends far, far, far before that diagnosis point. Sometimes it could be 10, 15, maybe even 20 years before diagnosis when some of these things start to show up. Now, one of them, excess daytime sleepiness. I mean, we've all experienced that. And apologies, it's the afternoon, so you may be experiencing that at some point this afternoon. But along with anxiety, depression, constipation, and hyposmia, which is our reduced sense of smell, these are just things that commonly happen as we age. Anyone who starts to age may experience these things at any point in time. So the problem that we really have is that these non-motor aspects that may present very early in the what we call the pathological process or timeline are actually very non-specific to Parkinson's, so they don't necessarily help us identify anyone early. The exception could be this REM sleep behaviour disorder. So that's where during our REM sleep cycle, instead of reaching a point of paralysis where our muscles will not move, what people end up doing is during their dreaming, they act out their dream. So instead of being paralysed and not being able to move, actually they can move their arms and their legs, and they do so. So instead of running the marathon and fighting off goats as you go, and just seeing that in your dream, you're actually running by moving your feet. You'll be moving your arms to ward off all the goats, and you're probably also vocalising as well. So there'll be a bit of yelling, get away goat, things like that. So there's many different causes or reasons why someone might have this experience, but it does seem to be very common in Parkinson's disease and related disorders, so things like multiple system atrophy, so MSA, or also dementia with Lewy bodies or DLB. And there are some theories that this is sort of the earliest sign of a degenerative process for some people. So there is a lot of work happening in this area, trying to identify people very early on in the process, long before they are diagnosed, because if we're going to get to the point where we want to stop the disorder or delay that diagnosis point, we're going to need to be able to give medications and drugs at this point to slow that pathological process down. So there's a lot of work going on there, but it's a work in progress. Okay, so we'll move on. So I'm just going to introduce you to the two cohorts that we have at NZBRI. So we've got our NZP3 cohort. It's a longitudinal face-to-face cohort of people with Parkinson's from that wider Canterbury region. It's been running for around about 18 years now, and we've had, we've collected data from over 540 people with Parkinson's and actually about 130 controls. I've got to check the actual number. So our control group's much smaller than our Parkinson's group. It does have a focus on those non-motor symptoms that I was talking about, and in particular the development of cognitive impairment, which we know can be a key factor for some people. So obviously our data collection includes cognitive testing. We've had historically a very strong focus on brain imaging, so MRI, but also PET scans. And as Amelia mentioned in the introduction, we do some biosamples as well, so things like plasma and DNA. Our second cohort is the NZPEGS one, so this is a nationwide cohort, so not just restricted to Canterbury, but all across New Zealand, and it's online. So people join online, we do our data collection online. It's cross-sectional at this point in time. We do have the option of turning it into a longitudinal cohort. We just haven't activated that part of it yet. And we've currently had 457 people with Parkinson's and 475 control participants complete the requirements of that study. Our aim is to get to 500, so hopefully by the end of the year we will be there. This study has a focus on factors contributing to the development of Parkinson's. So really what we're doing through these online surveys is we're asking people to delve into their memories and try and remember what happened in their life before they had Parkinson's, all the way back to birth. So we're looking at environmental and lifestyle factors that may put someone on the path to Parkinson's. And we're also collecting DNA from this cohort, and we do that via a saliva sample, which is quite easily done through the post. Okay, so just in terms of the things that contribute to the development of Parkinson's, we know that genetics is a very strong, or has strong involvement. So this figure just, it might look a little weird, but it is the easiest way to summarise the genetics. So along the bottom, this tells us how common the mutation in a gene is. So we start at rare on the left, through to very common on the right. And then on the vertical axis, it tells us its impact on developing Parkinson's. So small impact at the bottom and a large impact at the top. So this group up here in the red circles, these are the genes where mutations have been identified that cause Parkinson's disease. So we know that there is somewhere, depending on which population you're looking at, around about maybe 10 to 12% of cases would be called monogenic cases or genetic cases. So they're caused by a single mutation in a prescribed gene, and that's 100% probably why that person's got it. These two sitting in the middle, they look a little lonely. So they're, sorry, I'm just going to go back. So the occurrence of these mutations is very rare. They will often cluster in families because that's how genetic mutations obviously are passed down through generations, but they're rare. So we don't have many, we don't know of many of these people in our population. These two sitting in the middle, GPA and LARC2, these are more common, but they're not super common, and their effect or their ability to induce Parkinson's is not guaranteed, but it's also not minimal. So they're sitting there in the middle, and these are the two that have generated a lot of research interest because it's not difficult to find people with these mutations to form a study on, and because they are the proteins that these genes encode for have very specific functions within the cells, and they've been studied very well, and they're becoming the targets for the development of new drugs to treat the disorder as well. So a lot of interest around these two genes. And then in this blue circle here, we just have a big list of genes, And these are ones that, these aren't mutations per se in these genes, but they're common variations that we know occur in the population. And when you get thousands and thousands of samples together, you can determine that these variations occur more commonly in people with Parkinson's disease compared to those who don't develop it. And collectively, each of these single variations has a very tiny effect, but collectively, that gives us kind of our base genetic risk for Parkinson's that we all have. So across our two cohorts, we currently have information from 556 people with Parkinson's and 337 controls. And we've been able to look at many of these, not all of them, but many of them, to see how commonly these mutations are found within the New Zealand population. So collectively, between GBA, LARC2 and Parkin, which is this little one in here, we have about 12% of our Parkinson's population are having some variation identified in those genes. Most of these, somewhere in the range of about 8 to 10%, will be the GBA gene, because that's just the most common one that we find. There's 5% of controls that have cropped up with variations, and they are almost exclusively within the GBA gene as well. And that's not unexpected. I do just want to talk quickly about the pink one, which is up here, in this is going to cause Parkinson's group. So the neurologist at Auckland Hospital made some observations that they had a strange number of young Pacifica men coming in and getting a Parkinson's diagnosis in their late 30s and their 40s. And that led them to do some more investigations, and they've now discovered that there's a very specific mutation in the PINK1 gene, which is very prevalent within the Pacifica community, and it's contributing to people getting Parkinson's at an incredibly young age there. Now, I just want to, if we look at our list of genes here in the blue circle, and I said they all have a very little effect, and then you can add that up to give a baseline risk, if you like. We call that a polygenic risk score, so that just means we're taking multiple genes into account to calculate this score. This is not the full population that I mentioned, the 500 and the 300, this is a slightly smaller group, but this is what the risk score looks like when you compare the controls to PDs. And it's a Z score, so it's a standardized score, it's a little bit almost manufactured in a way, and it's designed so that the control average sits on 0, so that's why their average, which is the dark line, is sitting on 0. And you can see that the PD, or Parkinson's average, is just above that. It's sitting at around about 0.5. Doesn't sound like a massive difference, but it is significant when you do statistics on it. But there are a couple of other interesting things to note. So these are called violin plots, and the bulgy bits sort of tell you how many people are sitting at that level. So you can see the control group have this big bulge around 0, so most of the people are centered around 0. And the Parkinson's group doesn't have that bulge, so they've got much greater spread across the scores. And we can also see there's a bit of a tail at the top there, so they have higher scores. And these are probably people that have variants in that GBA or LARC2 gene to probably get them up quite that high. This is just a different way of looking at that, the same data. So instead of having our score on the vertical, we now have it along the horizontal, and this bulgy bit around the 0 is this peak here. And you can see that there's a definite shift towards those higher scores within the Parkinson's group. So that's what we would expect, but it's the first time we've ever been able to do this in the New Zealand population. And it will help us do further work around how does your genetic risk perhaps interact with your environmental or your lifestyle factors to put you on the path. So we're more likely to use this sort of collective score across many genes when we're comparing or joining that up with environmental exposures, as we call them. So just a couple of little summaries or little bits of information that are coming out from that NZPEGS study, which is really focused on that lifestyle and environmental aspects. So we're not seeing any increased risk of developing Parkinson's if you've lived in a rural area, which is something that has come out in many different studies across the world. We've done this twice now, so we've used administration data. We did it, we found no association, and we were like, there's a lot of missing data here, so we were a little bit sceptical. But when we get people to report all the different addresses that they've lived at throughout their lives, whether it was urban or rural, we're getting the same answer. So it doesn't seem as though rural living in New Zealand is putting anyone at an increased risk of having Parkinson's. We are seeing that there is some evidence that specific pesticides will increase the risk, which again we know. So you may have heard about paraquat as the main one. But we're only seeing a very small number of people actually reporting these exposures. So it does not seem that that is really a main driver for having Parkinson's in New Zealand. Yes, it'll be important for a few cases, but it's not the bulk of the cases. That's based on reported exposure, either through the occupation that someone's held, but also through, because we know a lot of people have associations with agriculture and farming outside of their occupation. So we try to capture both of those exposures. That does not exclude exposure through the food chain. So we know that paraquat is used on food crops, and that there may be residual levels in the food chain. It's obviously very difficult to measure that. So it doesn't exclude that possibility, but in terms of people that are being exposed to it and being exposed at really high levels, we're just not seeing that in the New Zealand population. Having a history of smoking and coffee drinking is more common in the control group than the Parkinson's group. We would expect to see these are well established associations, so it's just nice to know that the New Zealand population is acting the same as everybody else. In terms of head injury, so something else we know that obviously our brain is very delicate, and head injury is to be avoided as much as possible, as much as you can, really. But so a greater proportion of the Parkinson's population are reporting head injury that required medical assistance. So we're not really capturing I banged my head and I had a sore head for a couple of hours, but I didn't go to the doctor level. Obviously, males are more likely than females to report a head injury. That's not surprising if we consider activities and things like that. One of the key things which has been indicated for Parkinson's is the importance of this loss of consciousness at the time of that head injury. So whilst we were seeing that the Parkinson's group were more likely to report a loss of consciousness associated with the head injury, it wasn't actually contributing to an additional risk of developing Parkinson's in the data that we have. And I just wanted to touch on sleep. It's not really a lifestyle or environmental factor that puts you at risk, but it is an interesting one because we know our brain needs sleep. Sleep's very important to our day-to-day functioning, but it's also very important just for our brain in general. And we know that people with Parkinson's have disrupted sleep. So I talked about RBD before, so that being behavioural sleep disorder. So 45% of the people with Parkinson's are reporting having had it at some point, not necessarily currently experiencing it. So people don't generally experience it on a day-to-day basis. They might have short periods where it happens quite frequently, and then some periods where they just don't have it at all. So it's a bit of a, sometimes you have it, sometimes you don't. But 45%, which is quite a decent chunk of everyone, compared to 10% of our controls. Just in terms of a general sleepiness score, so I think there's only maybe 10 questions or something in it, and it just talks about different situations and perhaps how likely you are to feel sleepy or fall asleep. So again, our Parkinson's group is scoring higher, so we would expect that. But I thought this was quite interesting, is that the people who report having RBD don't have increased sleepiness. So although RBD is obviously happening during the sleep, and it seems to be quite disruptive in that you're being very active, it's perhaps not having any downstream effects on how awake you're feeling the following day. I would be really interested to see sleepiness scores from the partners of the people that have RBD, because it's probably their sleep who's being disrupted more than the person who's actually dreaming. This is my take a brief slide. So these are just some of the famous New Zealanders, oh not New Zealanders, famous people, but with all the New Zealanders that I can put in there, that have sort of publicly acknowledged that they have Parkinson's. I try to keep it updated, so I keep my ears out for the news, and whenever someone new comes up, I try and add them. So what I want to do now is just flip towards the NZP3 cohort. So this is the face-to-face cohort that we have. Some people come in, they just do one visit with us and then they disappear. We have some people who have been with us for that entire 18-year period. So we have a lot of information about them. And one of the questions that we've sort of always wanted to address is, what does Parkinson's do to our life expectancy? Do you actually lose years of life because you've got Parkinson's disease? And what we've done is we used our NZP3 cohort to do the first part of this work. And these are funny looking graphs, I admit. They're called survival curves. So you start at the top where everybody's there, right? Because everybody's still alive. And then as people die, that's when the line moves down and why it's a little bit jagged. So it's going at different times. So from NZP3, so these are males here and these are females. So we've had 275 males, and during that, when was our cut-off? I think the cut-off was only a couple of months ago. So during that almost 18-year period, we've recorded 138 deaths within that group. And for the females, we've had 127, and we've recorded 35 deaths. So you'll note there, which I didn't mention before, but obviously Parkinson's is much more common in males than females, at about a 2-to-1 ratio almost. So that's why our numbers are so different between the sexes. So what we've done for each of these groups is create a control group from the New Zealand population. And we've just roughly doubled the numbers. So we've got twice as many controls as we do Parkinson's. And what we've done is you can select people, obviously by sex. So we match males with males and females with females. But we select people that are born in the same year. So for every Parkinson's person, there'd be 2 people selected for them born in the same year and still alive at the age of diagnosis. Because what we want to do is we want to measure mortality associated with Parkinson's, not just life in general. So we need to make sure they get to that same age. And then what we can do is we can plot the age of death, so that's, we've got age along the bottom. And then we can see if these lines overlap, as they do here, or whether they deviate away from each other, as they do on the males. So what does that mean? What you do is you take the 50% mark. So when you've lost 50% of your population, what's the age between the 2 groups? So here we can see that our Parkinson's males are 80, just under 81 years at that point, and our control group are just under 84 years. And the key thing is that the 2 lines are completely separate and there's a white gap in between. It's a really unscientific way of telling you that that difference is significant from a statistical point of view. If you compare that with the females, they're completely overlapped. So what we're seeing is we're not seeing any, what we would call excess mortality in the females, but we are in the males. And it's around about 2.5 years. So males with Parkinson's might expect to lose about 2.5 years compared to their general population people of the same age in the general population. Contemporaries was the word I was looking for right there. So this is quite interesting because the effect is obviously not the same between males and females. But I do want to, we've only got a small number of females that have died. So what we obviously want to do here is we want to expand this out, not just to our local Canterbury cohort, but to the national population. So we do have a way of identifying all the people with PD in New Zealand. That's how we get our numbers that Amelia was talking about. So if we can identify them, we'd generate a New Zealand population control group and compare the age at death there and see if this, what we'd call a trend at this point, I guess with the small numbers, if that holds up for the whole population. The other thing that we want to do is to look at why do people die? So what is the cause of death? And the death certificate, so when someone dies, there's a death certificate and the reason for death is recorded. And there's sort of two categories. One is the primary cause, what actually went wrong that made this person die. And then there are contributors, as in the second category. And so what we sort of rely on is that Parkinson's disease is recorded at least in that second category. So we know that this person has had Parkinson's disease. They may have died of a heart attack, which is not directly related, but there was probably some contribution of Parkinson's. So we sort of rely on that sort of information being recorded. I can tell you now it's not always. But when we look again at this, we're still just looking at the NZP3 people. And when we look at that information, and I can say that we're not just relying on death certificate information here because we also get information from families at the time as well. So it's a bit of a mix of information that we're getting. So we can see, so we've got females here and males here, and then each colour is sort of a broad category for the cause of death. So we can see that the red is respiratory, and we can see that from this Parkinson's group it accounts for about a quarter of all the deaths, and that's compared to about 9% in the general population. And respiratory obviously covers off things like pneumonia, and in particular aspiration pneumonia, and I can tell you that that accounts for almost 80 to 85% of those deaths within that category. And we know that aspiration pneumonia is a prominent cause of death. The other thing to note too is on the vertical axis here, we've got the age. So these deaths are occurring relatively late, around about 80 on average. The next one here, which is Parkinson's NFD. NFD means not further defined. So we do find on death certificates that sometimes that's the only thing that's listed. Even though we know that Parkinson's alone doesn't cause death. There's got to be some other thing that's happened that's causing death. But we do find there's quite a large portion of deaths that only have Parkinson's listed on the death certificate. That's not helpful from our point of view if we're trying to determine what are the main causes so that we can inform the health system to keep an eye on these things. So it's a bit frustrating to have this category, and certainly, we're looking at 18% of all of the deaths have no further information. So it's our frustrating group, as we like to refer to it. In the green, we have cardiovascular. So here, and the females here. So apparently, I'm told, this is a very well-established effect. So if females die from a cardiovascular event, they do so much younger than males. So you'll see quite a big difference in the age between the males and females there. So that seems to be holding true in the Parkinson's group as well, and accounting for slightly less of the deaths in the general population as well. Cancer not super informative at this point just to have cancer, because obviously there's lots of different cancers, and we know that there are some cancers that are associated with Parkinson's disease. So again, obviously we're going to move this to the general the whole New Zealand population as well, where we get obviously many more data points. So I'm hoping that we'll be able to split this cancer group out and actually look at the different types of cancer, because I think there'll probably be some differences there between the Parkinson's and the general population. The cerebrovascular, which is largely strokes in that light blue, and then in the two sort of shades of purple we have organ failure and other. We have noted that in terms of this not further defined group, that often these people will have got to the point of having significant cognitive impairment or dementia as well, which is perhaps also contributing to death at that point, and maybe also why there's not more definition or more information on the death certificate. So I'm going to leave it there. That's the last one. First of all, so this is all the people at NZPRI as of maybe November last year, I think. It's the most up-to-date photo I've got. So lots of people there have contributed to the work that I've talked about today in lots of different ways, and also is a collaborative effort. So funders from New Zealand, from overseas, and obviously our host institutions as well. So yeah, I'll leave it there, and happy to take any questions.
Hi. Talking about the genetic side of it, my husband is in your NZP3 group, and so he will have his DNA tested for that. Now he is the third generation in his family that we know of to have Parkinson's, and he's got two sons. The question is, can they be tested?
Yeah, good question. So just so everyone, did everyone hear the question? Yeah. So we've got 3 generations where we know at least one person in each generation has Parkinson's, and the question is what does that mean for the offspring? And it's a very good question. It's not an easy answer because it's possible. Let's imagine that it's GBA. Okay, so let's imagine that the gene that's sort of saying that grandma, dad, and partner all have Parkinson's is because of GBA, just because it's easier to put a name on it. What we don't know is if that mutation or variant has been transmitted to the next generation, because it won't necessarily do it. And there seems to be, and this is what's complicating, I guess, our progress in this field, is that even if you've got the mutation, it doesn't mean it's expressed the same way each generation. So sometimes it'll be expressed quite strongly, and sometimes it won't be expressed at all. It also, if it's GBA, it's perhaps not strong enough to cause the disorder, so it actually also depends a lot on some of those environmental factors that individuals have experienced through life. And it's not unexpected to think that multiple generations in a family might be exposed to a similar or the same exposures over time. But yeah, you'd need sort of careful investigation with that particular family to really nut that out and to come up with a proper story. Parkinson's is just not considered to be a genetic disorder, so genetic testing of offspring just isn't really done unless it's one of those ones in the red area. There might then be an argument for it to be done at that point when you know that the mutation is definitely going to cause the disorder. But again, even if we know that, we can't do anything about it at this point, which is the other ethical conundrum. So, and it's really up to individuals. Do you want to know, even if you know you can't do anything to stop it. I mean, you can make some lifestyle changes and things like that, which might help slow things down, delay the diagnosis as such, but we're not going to be able to stop the disorder at this point in time. So yeah, there are some really big ethical issues around that. The other thing too is genetic counselling in New Zealand just isn't really a thing. We don't have a very strong genetic counselling service, and they certainly often aren't up to speed with this type of genetics. It's more the ones that are targeted towards children or maybe cancer and things like that. So I think there's a little bit of a gap in the health service there in terms of being able to provide the information and really good support to be able to make that decision as to whether or not you get tested. Is that all right?
Yeah, can I ask a follow-up question?
Yeah.
So if you were to go down that route and you found out that actually you do have the same gene, would that be helpful just in terms of watching out for the earliest symptoms, in terms of getting an early diagnosis? Because David's diagnosis was significantly delayed because he was misdiagnosed for a long time. So I just wonder if early diagnosis is helpful significantly in terms of progression of the disease.
Yes and no. So, I mean, early diagnosis is good in terms of understanding where you're at, planning your future, things like that. But again, we treat the symptoms currently, so we don't have anything that's going to change the course of the disorder, unfortunately. That's obviously what the whole field is working towards, but it's still a wee way off. So, I mean, I'm all for early diagnosis. I don't think you would need to know that there's a genetic mutation there to keep an eye out for it, because you guys know what you're looking for. The trick is to advocate. If you are seeing things, if you're seeing signs, to advocate with the medical doctor who's sitting in front of you, who might not be so familiar, particularly if it's at a younger age, to advocate for those children and to get a diagnosis as early as possible. So yeah, I think you can achieve what you're sort of asking, but without the genetic information, because you've got a strong family history, so that should ring alarm bells right from the get-go anyway. In terms of the treatment, we're still just going to treat the symptoms that are present, because there's not a lot we can do to change the course of the disease at this.
Another question is,
actually, this lady's follow-up question was just about answer.
Oh, okay.
From your first chart, I've had a lot of those symptoms for many, many years. It was the REM, sleep disorder. I've got sent to a neurologist who said this may lead to Parkinson's in the future, but nothing was followed up. And then it's only my daughter that's put together this year all the different things that I've experienced, sent it to the doctor saying, I think this needs to be investigated. I think she needs to go and see a geriatrician and get assessed. So my question was, if I had been diagnosed way earlier, would it have helped me to get on the medication earlier?
Some of the symptoms are like 20 years old, the loss of smell.
Yeah, so loss of, I mean, like I said, some of those non-motor symptoms in particular are just non-specific, unfortunately. We know that they are common within the Parkinson's population, and we know that it certainly seems to, if you turn up with your loss of smell, increase your risk. It doesn't guarantee it, unfortunately. And the medications, no, I don't think so. So like I say, I don't know if you'd never really considered actually giving levodopa at that point in time. You might get a very small benefit, but it's probably not necessary until you've got those motor symptoms. So, the levodopa, the main medication, is replacing the dopamine in the brain that you've lost. So what we really need is something that we can apply in that early period. So if you have your REM, behavioural sleep disorder, you go to your GP and you're like, Mary's acting out her dreams and going crazy, and we can give something then that might slow down the pathology that's happening in the brain. So instead of getting diagnosed 10 years later, you might get diagnosed 15 or maybe 20 years later. So we just need to try and stretch out that timeline. That's the most likely scenario at some point in the future, rather than we're going to knock it on the head and we're going to stop it completely. That's going to be a bit tricky, but dragging out that timeline would be the goal. Because what we want to do is we want to try and increase the length of time that people have got good quality of life. And good quality of life is less symptoms, right? Less severe symptoms. So if we can drag it out, that would be very beneficial. So we're not currently at the point, I think, where we can sort of advocate for treatment at that point, because it's probably going to be a medication or a drug that we just don't know about yet.
And with the levodopa,
Yeah.
it's taken me a long time to be able to increase that, so that I'm not tired, no strength, I can't go for a walk or do much at all. I'm only up to 1.5 pills and I need to get to 3 because I'm going to go backwards because I'm feeling so terrible, worse than I felt before I've been on. So does it normally take a long time to?
That's a good question. I'm not a clinician, sorry. Interesting that you mentioned lots of things, but you didn't mention nausea. So nausea is one of the big things that people have trouble with. Yeah. Okay. Yeah, I don't know. I guess we're all different, so maybe you're at the far end where it's just taking a little bit of time. Yeah, I just persevere if you can. Yeah.
What cancers are associated with Parkinson's disease, and are the associations strong enough to warrant screening?
That's a good question. So melanoma is one. So people who have melanoma are at increased risk of getting Parkinson's, and people with Parkinson's are at increased risk of getting melanoma. So that's a key one. And I think there are some associations with breast cancer and also prostate cancer as well. So that's slightly more common in the population. I would say I don't know the numbers enough to be able to say yes, we should be screening. I think we should be screening for cancer anyway. Like if you're in the sort of the age range that's at risk.
But if melanoma is a risk, we should be at least be advised to avoid sun exposure.
Yeah, well, I think we're in New Zealand, so we should be anyway. Yeah, I don't know. I think it's one of those things where I think when you look at large data sets, you see it, but when you're looking at an individual patient in front of you, it's probably hard to think. There's lots of other stuff going on, so it's difficult to think I should send this person for screening. Yeah.
The mortality differences between male and female, does that mirror studies that have been done overseas in the same area?
That's a good question, and I must admit I've not been the one who's done that analysis, and I haven't looked into it too much at this point, but yeah, that'll be one of the things that we're looking at to see if there is that difference. But I'm a little bit concerned about our female group. There is a very low number of deaths at this point. So I'm hoping once we get to that full New Zealand population level, I mean, it would be really nice if it did show the same sort of thing, but I think that we'll have to wait till that point before we can really sort of hang our hat on it and say that this is definitely what's happening.
One more in here? No?
Oh, sorry.
Is there any relationship between Parkinson's and the familial tremor? I know it's called something else.
Oh, essential tremor.
Essential tremor. Yeah.
Yes and no. So there's the misdiagnosis issue, right? So sometimes.
Self-diagnosis.
So even though they're quite different tremors, sometimes there's a little bit of ambiguity as to whether it's essential or whether it's Parkinson's. So there is a little bit of a misdiagnosis issue there. Again, I think if you look at really large numbers, people that come and are diagnosed with essential tremor are at increased risk of going on to develop Parkinson's. Now, whether that means essential tremor turns into a Parkinson's tremor, or whether it means maybe it's been misdiagnosed early on, I'm not aware of, I don't know the ins and outs of that. So yes, I would say there's an increased risk, but again, it's not guaranteed.
No.
Certainly not guaranteed.
I have a sister with that, and she's had it, she reckons, since she was about 12,
Mm.
and she's now late 70s.
Yeah.
She's still the same. She's fine.
Yeah.
It's not fair.
I read a study a while back on the post-mortem results of people who had died with Parkinson's, and it suggested quite a strong relationship to brain injury. I think about 80%, as I recall, had some form of non-major brain issue of one form or another. Do you know if there's any research looking at the likely cohorts that would suffer something like that? I'm thinking particularly rugby players and boxers.
Yeah, so this is a really big, I mean, this is a huge area of research, and it's really just blowing up in probably the last 6 years or so. Not with Parkinson's as the end, but that dementia, right? So special type of dementia which people get if they've been involved in activities where you get head knocks, but not proper head injuries, if you will. Yeah. So these are not injuries per se, and they're certainly not losing consciousness. They're just generally sporting activities like rugby league, ice hockey, American football, where there's impact. So repetitive impact, because every time you get a tackle, your brain's moving, right? And your brain doesn't like that. So there is a whole set of pathology which seems to be associated with that, which seems to be separate from an Alzheimer type dementia, even though there are obviously similarities. Hits people young in many cases. So let's think about Carl Heyman. So he's an ex-All Black who's come out and publicly acknowledged that he has dementia, which is probably due to his rugby career. There's obviously big lawsuits in NFL and through the, I think it's the English rugby players are leading one as well. So potentially at some point we could find that there's something similar leading to Parkinson's in terms of, instead of getting those actual head injuries that are recognised as head injuries, but those lower grade sort of damage may over time accumulate, and for some people might lead them down to the dementia route, and for some other people might lead them on to Parkinson's. So yeah, it's not something that I've seen yet, but it wouldn't surprise me if it comes in the future.
Paper had an interesting comment to make that they found a significant number of people that had such incidents 10-ish years prior to development of Parkinson's syndromes. So there seemed to be some kind of long lead in if there was actually causal.
Yeah. So often with these things, it's accumulative. You know, we often, you know, on average, someone's going to, the average age of diagnosis is early 70s, early to mid 70s. So it's a bit later than most people think. So it is an accumulation of damage over time that is probably leading to it, rather than an impact that's happening or an exposure that's happening and then, you know, the next day, the next month, the next year. Takes time to, obviously, your brain's very resilient, and I think that's one of the reasons why there's such that long lead-in time. So your brain can actually accommodate very well. So if there is damage, it fights to keep functioning normally, and it's just once it reaches that point that it can't fight anymore is when we start seeing the symptoms. So yeah.
Well, thank you so much, Toni. You know, it's completely, I find it completely heartening that the research for New Zealand is happening in New Zealand, and that we're not having to solely rely on international studies. And it's thanks to the researchers who are doing it, to the institutions that are enabling it, like NZP3 and University of Otago, Christchurch School of Medicine, University of Otago School of Medicine at Christchurch. But and you know, and the willing participants as well, such as you people in the room. One of the things that stood out for me, which made it absolutely crucial that we were doing this research in New Zealand, is that pink gene with Pacifica males. I mean, we're unlikely to see that in other places in the world so readily. If we don't do it here, we don't find it here, and we can't support those people here either. So thank you.
Yeah, and that is also leading to the neurologists from New Zealand going to the Pacific Islands as well, and supporting the neurologists there who are obviously seeing these cases as well. So yeah, I think it's a really exciting area, that one.
And on a sort of more personal note, I'm absolutely heartbroken that my son plays rugby. And he said 7 head knocks. I allowed 7 head knocks in my life. I'm like, seriously, Theodore, 7? He's on number 6, so I'm like one head knock to go. So please join me in thanking Toni for coming and sharing the research